Good evening, this is Joshua, and for this project, I'm assigned the melanocortin 1 receptor protein, MC1R, and the arginine number 67 mutation replaced by cysteine, R67C, affecting beach mouse coloration. Beach mouse fur coloration. So the normal function of the MC1R protein is to, when stimulated, produce a lot of nucleotide CAMP used for intracellular signal transduction, which leads to the expression of at least 4 genes, CTYR, TYRP1, TYRP2, and P. As a receptor protein, the MC1R becomes stimulated by receiving a signal, where increased stimulation leads to increased eumelanin production or darker hair. The protein is normally found within the cellular membrane. The MC1R protein is found in hair follicles, skin, like the epidermis, and eyes. Lastly, MC1R proteins are found in any organism containing melanocytes, which are plants, animals, and filamentous fungi. So the type of mutation on this protein is it causes the protein to become less active. Essentially, the cysteine right here that replaces arginine at amino acid number 67, indicated by the end of this model here. I did not continue past, I only can continue past up until number 67 amino acid. I only have transmembrane 1, but on the full protein, there are several. In fact, there are 7 transmembranes on this MC1R protein. I only have 1 here on this model, which is one of our limitations here. So it only goes up to our 67th amino acid. All right, back to the explanation. So essentially the cysteine that replaces or displaces arginine in the mutated protein causes the protein to be unable to bind to alpha MSH, which is a melanocyte stimulating hormone. The mutated MC1R protein affects its ability to regulate nucleotide CAMP necessary for intracellular signal transduction pathways adequately, to regulate nucleotide CAMP adequately, leading to less stimulation of the protein and ultimately less activity or normal response. And this normal response that we're looking for is eumelanin production, so the stimulation of TYRP1, TYRP2, and P. Now, the lack of stimulation favors the intracellular signal transduction pathways that result in the production of pheomelanin, the pigment for lighter skin. So less CAMP will lead to less stimulation and therefore less eumelanin production and more pheomelanin production here. Now, so when we're looking at the amino acid chain for arginine, we know that the side chain for arginine consists of 2 amine groups. Also, the side chain is polar because it's charged, and the side chain is positively charged. And that's really important because that makes it basic. It makes it basic. And so it's going to be a proton acceptor when put into a solution, an aqueous solution with water. Well, from a biological perspective, it would be in the body where there's a high water concentration. It's hydrophilic. And arginine is considered larger than most amino acids, with a generally long side chain composed of mostly CH2 and amine groups. Now, in arginine, one of the amine groups branching out at the end of the side chain is positively charged, which means it acts as a base and likely accepts H+ ion in water to form NH2+ from HN. Furthermore, the other amine and NH groups are also hydrophilic. It will be attracted to water and form hydrogen bonds. Hydrogen's hydrophobic side, contrastingly, is composed of nonpolar CH2 molecules that will not interact with water and interact with other hydrophobic side chains. Now, the main functional group in the side chain of our mutated side chain is cysteine, right here. And cysteine is indicated by TGC, and then, it's TGC. And cysteine's side chain is hydrophilic polar, consisting of only a sulfonyl hydrogen group and 1 CH2 molecule. Cysteine's side chain is polar and is also neutral, and its side chain is considered smaller than most amino acid side chains. Like arginine, cysteine can participate in hydrophilic interactions. However, cysteine does not have a charge, which is required for stimulation of the M1CR receptor protein. That's why less stimulation leads to less eumelanin production. The pH of cysteine, for this reason, does not exist as opposed to arginine, whose pH is considerably basic. Now, the mutation that displaces arginine with cysteine at amino acid number 67 of the MC1R protein affects how the protein responds to alpha MSH. Well, does not respond, which affects its ability to regulate CAMP effectively. This causes an inadequate response from the MC1R protein, favoring the signal transduction pathway to express the gene necessary for pheomelanin production, not eumelanin production. This change is seen phenotypically in house mice with fairly toned fur, low eumelanin production, compared to house mice with darker toned fur, high eumelanin production. Now with this model, like I explained earlier, the protein does have 317 amino acids and 7 transmembranes. This only pictures 1 and goes up to the 67th amino acid and the 199 to the 201st monomer here. And so this model also does not have many bead variations to account for the 19 different types of amino acids. There's only ranges from colors from orange, purple, green, lime green, and then we also have some red, I believe, in there, but it's difficult to see. And so each bead is similar on both of the proteins here, except this 67th one, where one is yellow on the to represent arginine and one is blue to represent cysteine. And I showed it here in marker that it's the 199th one here where the cytosine is displaced by thymine, and that causes the change in amino acids. And it's just to show how large of a difference just one mutation can cause. It causes it affects the whole ecology of beach mice when they're fur. They're going to be attacked much more in darker areas like wooded areas if they have lighter fur, and so it has a great effect on the ecology. And now this model is not positioned on a bilayer. The MC1R protein is usually on a bilayer, and it is and this is just on a table and it's laid flat down. It's usually positioned on a bilayer, and those are our 3 limitations here. Thank you so much. Goodbye.